Hello, my name is Salik and I am making this video for Khan Academy's talent search for India. So basically this video is going to be of 10 minutes, and in these 10 minutes I'll try to explain the structure of DNA, that is Watson and Crick model. So without any further delay, let's begin this video. As we know that DNA is the polymer of nucleotides, that is DNA, we can say DNA as well as RNA, both are nucleic acids. DNA and RNA both are nucleic acids, and nucleic acids are consisting of nucleotides. There are two terms that you might have heard, that is nucleoside and nucleotide. Nucleoside is basically nitrogenous base pentose sugar. Nucleoside is basically nitrogenous base, that is purines and pyrimidines, adenine, guanine, thymine, cytosine plus pentose sugar. While nucleotide, in addition to nitrogen base and pentose sugar, nucleotides also contain phosphate group. Let me write nucleotide by NT and nucleoside by NS. Nitrogenous base plus pentose sugar plus phosphate. So phosphate is the addition factor that is present in nucleotide. So nucleotide have this thing in addition, and DNA is the polymer of these nucleotides. So one nucleotide will contain a phosphate sugar, which is a ribose sugar which contains 5 carbon. It will contain a phosphate and it will contain a nitrogen base. Nitrogen base, sugar, phosphate. This nitrogen base can be anything. It can be adenine, it can be guanine, it can be thymine, it can be cytosine. It can be any of these four. And the second thing that you have to know is that adenine and guanine both are known as purines, and thymine and cytosine both are known as pyrimidines. These two are known as pyrimidines, these two are known as purine. And in DNA there is a base pairing that occurs, that is two strands are attached to each other, as you might have seen in this video or any DNA structure that was given by Watson and Crick. What they said is that if we open this DNA and if we remove the twisting thing of this DNA, that is, we will stop drawing it in the helical form, but we will draw in a straight form. That is, it will be looking like a ladder. There is always a strand, or there is always a bond that we can see, this bond. This bond is between one strand and the other strand, and basically here there is a nucleotide, there is a nitrogenous base, and here also there is a nitrogenous base. So this bond is between two nitrogenous bases. And what Watson and Crick said, that this bond will always be between purine and pyrimidine. It can never be between two purines and it can never be between two pyrimidines. It will always be present between purine and pyrimidine. And which purine binds with which pyrimidine, that we have to know. That is, adenine always binds with thymine and guanine always binds with cytosine. This we can remember through an acronym that is known as apple on tree and car in garage. Apple on tree and car in garage. This means A always binds with T and C always binds with G. Okay, and always you have to remember that between adenine and thymine there will be a double bond, and between guanine and cytosine there will be a triple bond. So this is complementary base pairing that you have to know. Alright, so this was something basic that you had to know about DNA, and this model was given by Watson and Crick. So Watson and Crick basically gave their model in 1953, and before that in 1953 Watson and Crick gave their model, that is Watson and Crick's model, double helical model, or we can also call it. So in 1953 they gave their model. In 1962 they were awarded Nobel Prize for this model. But one thing you have to know is that Watson and Crick basically all their structure and research was based on the earlier model. They modified the earlier model that was given by Wilkins and Franklin. Wilkins and Franklin, what they did is that they studied the DNA molecule with the help of X-ray crystallography, and on this model Watson and Crick modified their own model, and it was based on this. So in 1962 the Nobel Prize was not given to two people. It was not given to two people, but it was given to three people. In addition to Watson and Crick, Wilkins was also awarded Nobel Prize. This thing is that you have to remember. The Nobel Prize that was given in 1962 for the structure of DNA was given to 3 people, and in those 3 people Wilkins were also included. So this double helical structure is something that you knew, that I tried to make you understand. And now the thing that you have to understand is the configuration of the DNA. That is, what is the basic configuration of DNA. As you can see in this diagram that is drawn by me, the DNA consists of various twists. You see, if I say that it is simply like a ladder, and we will twist this ladder, and it will be present in twists, and these twists are known as helices. Helix. One twist is known as helix. For example, from here till here there is one twist, and from here till here there is another twist. Okay, and what Watson and Crick said, that this one twist or one helix, the length of this one helix is 34 angstrom, and as you know, 1 angstrom is equal to 10 raised power minus 10 meter. So the one helix, the length of one helix is 34 angstrom, and in this one helix there are 10 base pairs that are present, 10 bondings that are present. So if we say the distance between 2 base pairs, how much will it be? 34 divided by 10, which is 3.4 angstrom. So the distance between 2 base pairs in a double helical DNA is 34 angstrom. Alright, and the diameter, if we say this ladder, if we twist this ladder, there will be a constant diameter that will be present of this helix. So that diameter is 20 angstrom. This is something that is said by the Watson and Crick model. The diameter that is of the DNA is 20 angstrom. Alright, and as we know, there are many types of DNA, which will go into detail in any other video. In this video, I will briefly say there are mainly 4 types of DNA, that is A, B, C and D. And what we most often see and what we most often study is this bDNA. And its structure I have explained above, which contains 10 base pairs in a helix. aDNA contains 11 base pairs in a helix, cDNA contains 9.33 base pairs in a helix, and dDNA contains 8 base pairs in a helix. This thing that you have to understand. And what we study often is the bDNA. And in whole of one human DNA, there are almost 6.6 into 10 raised power 9 base pairs that are present. This thing is something that you have to remember, and there are many objectives in NEET NEET as well as in AIIMS that come on these types of facts, and you have to understand that. And the last and the most important thing that was said by Watson Crick is that they are the 2 strands, the complementary strands that are present in DNA that are attached with these hydrogen bonds are anti-parallel to each other. That is, if one strand, if we open this DNA, if this one strand is 5-3-, the other strand will be 3-5-. Now what is this 5-3-3-5-? This is a very elaborate topic, and if I try to explain this briefly, the carbon of sugar in the nucleotide, as I said, sugar is like this, here is phosphate and here is nitrogen base. The sugar, the number of carbon in sugar that is present here, it is the 5th carbon that will be present in the top side, and the sugar that is present here, it will be the 3rd carbon that will be present in the top side, and this carbon will be inverted. This chain, if we invert this chain, this 5 carbon will be present here and this 3rd carbon will be present here. As I said, this to make you understand this concept, I will require a complete video. So if I put this briefly, this is the way I can put it. So with this, I tried to explain a little bit of DNA, its structure, that was given by Watson and Crick. I hope you liked it, and thank you very much for giving us this opportunity of trying to work with Khan Academy, and I hope to see you again, and I will be awaiting for your email. Thank you very much and good luck.